All right, I'm just going to scroll through one more. Again, you are at the ALS TDI Town Hall, where we'll be talking about the ALS Research Collaborative, ARC. So welcome. Okay, we're at 3:04. So just to make sure that we're able to continue with or cover probably a lot of content, I'm going to start by introducing myself. My name is Carol Hamilton. I am waving my arm in front of a virtual background. I am the Senior Vice President of Development at ALS TDI. Thank you again for joining us. Before we start, I want you to all take a look at that slide. On the bottom are our sponsors that allow us to present these educational programs to you, including the town hall. So we have Cytokinetics. Most of these names are known to you: Biogen and Amylyx, Mitsubishi Tanabe, Apellis, Novartis, and of course the wonderful InSource. So thank you to all of our sponsors in helping us have the wherewithal to bring all of this information forward. Everybody is going to be put on mute except for a very select few of you. We are then going to, you can unmute at the end for questions. We will open everything up to questions at the end. If in the interim you have a question and you are capable of typing, please go ahead and put it in the chat. I have some friends on here who are going to help me monitor that. And some of those people I'd like to introduce now. Could the development team at ALS TDI please wave their arms around like crazy people? Thank you. Michelle Dupree, Glynis Murray, Melissa Payne, and Courtney Mish. These are the people who are going to reach out to you afterwards, if you don't already have a relationship with them, to ask you how we did and to get feedback on how we can be better and topics we can cover in the future. You will also get a questionnaire from our marketing team, an email coming from ALS TDI. So if you have something horrible to say and you're scared to say it to one of those 4 people, please still say it in there so that we know how to be better. From there, I'm going to turn it over to our Manager of Science Communications, John Gang, to hit it up from here.
Thank you, Carol. As Carol just said, my name is John Gang, the Manager of Science Communications here at ALS TDI. And just to run through the agenda for today, it's pretty simple. I'm going to do a brief introduction to the ALS Therapy Development Institute for those of you who might not be completely familiar with us. Then we are going to hear from Alan Premiseri, our Senior Manager of Clinical Operations at ALS TDI, about the ARC study and why you might want to participate in it. And we'll also hear from some ARC participants, and then we will open things up to your questions for a town hall discussion. So just a little bit about ALS TDI. Our mission is to discover and develop treatments for everyone with ALS. And ALS TDI was founded 25 years ago by the family of Stephen Haywood. You can see them down there in the photo on the left. Stephen Haywood is in the orange shirt in the back. Stephen was diagnosed with ALS at the age of 29, and when his family found out that there were no treatments available for him, they decided to do something about it. And so that was our first office in their basement in Newton. They founded us as the first ever nonprofit biotech company. And 25 years later, we are the most comprehensive drug discovery lab in the world focused solely on ALS. We have a state-of-the-art laboratory in Watertown, Massachusetts, and we are still funded by the community. We put 87 cents of every dollar we raise into that lab to find effective treatments and cures for ALS. And Stephen passed away in 2006, but as you can see in the picture on the right there, the gentleman in the blue shirt in the middle, that's Stephen's son in our lab with some of our scientists. So you can see this is still a family affair, and the community is still part of everything that we do. Now, at ALSTI, how our mission works is we aim to be the drug discovery engine for ALS. And that means we know ALS is a complex disease. We know no two cases are exactly the same. And so we know it is going to take many treatments to end this disease. So that means that we do the rigorous preclinical research that is needed to fill the ALS clinical pipeline. And we do this by working in our lab to discover and invent new potential ALS drugs. Then we partner with others to help ensure that those drugs advance to human trials, unlike another more traditional biotech company that might follow those drugs through trial and then bring them to market. This allows us to go back to the drawing board and keep looking for more treatments and to continue to do this. And we won't stop until everyone with ALS has effective treatments. And over the past 25 years, we have rigorously tested over 550 drugs in animal models of ALS, which is more than any other ALS research lab in the world. We were the first nonprofit biotech to invent a potential ALS treatment, tegoprubart, and bring it from our lab through FDA review and into clinical trials, where it completed a phase 2A study in 2022. We also launched the ALS Research Collaborative, or ARC, which is the study we'll be talking about today, where anyone with ALS can contribute their data to help accelerate ALS research. And we also developed the ARC Data Commons. That is a cloud-based platform that shares the data that you contribute to ARC with researchers all over the world. People from any institution can access it, and this means that when you contribute data to ARC, it doesn't just move forward research at ALSTI, it is moving forward research in the entire space. Another thing we want to make sure everybody knows about is another service that we introduced last year. It's called the ALS Trial Navigator. And this is a set of tools that can match you to trials and help you make informed decisions about what you might want to enroll in. And this platform allows you to search trial anywhere in the world, from the US to Europe to any other ALS trial that is currently going on anywhere in the world. It allows you to get tailored recommendations by answering a questionnaire, which will give you a personalized list of trials tailored to your needs and preferences. Also allows you to create a personal portal where you can save trials, make notes, and bookmark ones that you're interested in. And along the way, you will learn important terms and concepts that will help you make informed decisions about participating in ALS research. And with that, I'm going to hand things over to Alan to talk about ARC.
Thank you, John. I'm going to switch over to my screen. Can everybody see that okay? I see a thumbs up. Awesome. Well, I see a lot of friends on here, so hello, friends. I can't say who you are, but you know who you are, so hello again. And some new faces as well, so thank you for joining. And for those watching the recording, hello. Thank you for watching. So I'm Alan Premiseri. I'm the Senior Manager of Clinical Operations at TDI. I've been with the company since 2013, and I switched over to the clinical operations side around 2020. So today I'm going to give you an overview of ARC along with some other things. We'll start by showing you the agenda. If my slide switches, here we go, there we go. So as I always have to do, I have to give an ARC overview for the people who need a refresher or aren't familiar with the study, just so we can have sort of an up-to-date information on everything. I'll touch on some updates and collaborations. There's kind of a lot going on, so I'm not going to go deep in on anything, but if there are questions, like put them in the chat. My email is probably accessible to most people, so feel free to email me directly if you're curious about any of the projects. And then we'll have some participant perspectives. I have a couple of very generous participants who are willing to give a little bit of their time today to speak about their time in ARC. I'm prone to going on tangents. I'm going to try to go through this smoothly so we have time to do everything at the end, because there's kind of a lot of ground to cover, but as I said, if there's questions, put them in the chat. All right. So I wanted to start just by framing ALS TDI's research roles from the sort of the highest level, because we're only going to be talking about one of the programs today, which is ARC. But for those who don't know, this is the drug development pipeline laid out from basic biology, fundamental research, all the way to getting an approved therapeutic. And ALS TDI, most of its history spent in the left side here, what we do in the lab, which is fundamental research, drug screening, using animal models or cell models, selecting potential therapeutics that we found in those drug screenings to move eventually into IND enabling studies. So we do a lot of this. We do clinical research through the ARC study, which we take findings from and use that in our decisions in the lab to help guide this part of the process so that we can make this more efficient and more informed and direct our funds a little more appropriately. And then across everything, we work with partners. So we're a very collaborative group. We're pretty flexible in terms of working with people with specific expertises or other general sort of scientific areas of research. So we do a lot of collaborating. So I just wanted to say that up top as we go in to focus on ARC. And another sort of framing device I wanted to put up front here is covering the difficulties in ALS research, not just for us, but as a whole field. There's still large gaps in our biological understanding of ALS. So this is like mechanism of actions, what pathways are contributing to the disease, what biomarkers trend with disease. We are making way, and we're finding specific maybe pathways or specific biomarkers that can be attributed to certain people or certain progressions of ALS, but there's still so much we don't understand on a molecular level. There's a diverse set of symptoms and progression patterns. So we all know ALS is very heterogeneous in how it presents. This affects how we assess potential therapeutics in clinical trials. So I guess a good example, although it's very specific, is tofersen, which got approved for SOD1 ALS, which is a specific subset of people with ALS. There may be other subsets, and they might not be genetic causes. Perhaps there's a treatment out there that's specifically for people with limb onset ALS or bulbar ALS or fast progressing ALS. We don't know enough about who fits in what categories, how to categorize people. And that affects how we assess treatments. And then, of course, there's the different biological factors, which ties into the previous point, and environmental factors. We don't know sort of what's outside the body that might be affecting somebody's progression or even triggering it in the first place. Two people may have similar living situations, and for whatever reason, they progress differently in their ALS. We don't know how environment plays a role into ALS, at least not well enough yet. And then the last point I wanted to address is that there's still a need for sensitive measures of ALS progression. Many of you know that the ALS functional rating scale is the gold standard. It is the primary endpoint in most clinical trials for ALS. But as we're doing our fundamental research and our basic biology, we still get hamstrung by the question of how does this biological response pair up with a clinical presentation? And we can do that research, and there has been good, like, for example, neurofilament has had a lot of research done with ALS. But if we're not measuring somebody's symptoms properly and sensitively, are we truly understanding the biological effect of whatever pathway is being affected or whatever protein is doing harm to somebody in their ALS? So those are the three sort of difficulties I wanted to highlight. And I want everybody to keep these in mind as we go through ARC. Hopefully by the end of the overview, at least, you'll see why ARC sort of is addressing these points. So the ALS Research Collaborative, or ARC, is a natural history study I think this is more of a well-known term now. It's a study where you just collect data from people in a given disease indication as they're living their life, to understand more about that given disease. And in this case, it's ALS. And our aims, and ties in with the previous slide, are to find new targets relevant to ALS subtypes. So maybe if there's a genetic marker that we find that we can inform or categorize people, maybe this gene causes this progression of ALS. We want to know that. Develop new models and test potential therapeutics. So this ties into the idea of us bringing that information back into the lab. If we find something like a protein or a pathway that's affected, we can tell our bench scientists, maybe this is worth developing a cell model for or an animal model for. So that quick translation from the clinical research back into the lab and is another big point of why we run the ARC study. And then to discover or validate biomarkers to monitor progression. This is a little bit more to help the clinical research community as a whole. If there's anything we can do to help develop something that is a sensitive measure of ALS or help validate a biomarker, because we have so much data from so many people, we're in a position where we might be able to help others reinforce their findings and hopefully get those findings implemented in clinical trials. This is my fast facts slides about ARC. We launched in 2014, so 10 years now. It's been going. It's completely remote. It runs indefinitely, so we don't have any intention of stopping it. People can sign up and opt out at any time. I think most people tend to stay in the study for somewhere between 6 months and a year. And we crossed 1,000 participants enrolled recently. So that's great, and that's awesome, and we're very grateful for it. And so now we're aiming for 2,000. And last year, at the end of last year, we started formally enrolling asymptomatic gene carriers. The only difference between the people who are gene carriers and the rest of the participants in the study is that they just do all the tasks less frequently. And hopefully they don't, convert to developing ALS symptoms, at which point they would just join the study as a normal participant. If not, we'll just do less frequent data collections. All right. So people sign up for ARC at arc.als.net. There's a little 4-minute video there that should give anybody an overview if you're curious, after this recording of what the study entails and need a refresher. There's a pretty detailed enrollment process as far as describing like what's involved. Hopefully by the end of all that, you should feel comfortable signing the informed consent. Because this is all remote, you're not coming to talk to us in person, although you can reach out if you have questions. So there's a nice enrollment process there to get you up to speed on what the study entails and all the obligations and really not obligations there are as part of the study. And once you're enrolled, you'll get access to a portal that looks like this, where you'll have access to your data that you've submitted, seen here on the left in these menus, and then a dashboard where it'll essentially keep track of the things you have to do, the things that are owed. You'll get email reminders as well to help you stay on task. One point I like to emphasize is that it is not required for everybody to do every part of this study. So if that is a concern, you can sort of a la carte pick, I'll do the FRS and the surveys, and maybe I'll do the FRS, monthly, and that's it. And that's fine. We're really grateful for all the data we can get. It helps that the study is remote and that it doesn't have an end date in mind. So we're just trying to collect as much data as we can. The 5 main tasks of the study are listed here on the left. I'll go through them a little bit quickly, but I think they should be familiar to many people if you've sort of seen other non-interventional research. So the ALS Functional Rating Scale is, as I mentioned, it is the gold standard of measuring disease state and progression in the clinic and clinical trials, and really in just basic clinical research as well. It is a 12-question questionnaire that assesses a patient's basic daily functions. So like speech, salivating, swallowing, there's like cutting food, walking, climbing. And they assign a score from 0 to 4. It's an integer score, where 4 is normal function and 0 is essentially loss of function. it has been the gold standard for a very long time. I think it's a prevailing thought in the community now that there are significant flaws with it. It's a bit subjective. You can't pick anything in between the numbers that are assigned there. So if you're feeling a 3.5, you won't see it. So there is some sensitivity loss there. It begs the question of if we're capturing enough information, but it holds up well enough that it's still being used. So we include it because we need to, if we're developing new techniques to measure ALS, we have to compare it to this. And it's not something we should necessarily get rid of, but maybe we should supplement with other measures. So this is sort of our anchor for measuring. We have surveys, a whole bunch of surveys that capture a wide range of topics. So I've listed them here, but family history, lifestyle, like smoking habits or injuries, vigorous exercise, where you've lived, where you've worked. And then you can keep track of your medication, supplements, and clinical trials that you've participated in. If that's helpful, some people will log their medications. If they're doing the FRS monthly and maybe doing like speech recordings, they'll be able to try to line up and see how this treatment regimen might be affecting their scores in the other areas. But the other thing here that's important is that environmental risk factors are an underdeveloped part of ALS research. And we want to know if there are things in the environment that may be linked to how somebody's progressing or when they got their symptoms or what symptoms they were when they first got them. So this is going to be sort of one of our bigger projects in the coming year that is hopefully going to give back to the community in an area that hasn't been explored well enough. Speech tracking and voice recording involves a monthly phone call. It's an automated phone call where participants basically hit like a call me button. They have a script in front of them to read 5 random phrases each month. So they'll take the call, read the script in front of them. We'll take that. This is just a computer. I don't know. We'll take the recording, store it in our servers. And with the help of Google, who developed a machine learning model a few years back, we take that recording, turn it into a spectrogram, which is what you're seeing here, of somebody saying like 5 phrases in image form. And their model basically looks at the image, cuts it up into a whole bunch of pieces, and gives each little piece a score from 0 to 4. And it spits out in the end like a predicted score from 0 to 4, where 4 is again normal and 0 is complete loss of function. And in the portal, participants will be able to see those scores displayed on a graph like this. As I mentioned with the FRS, you can't pick anything in between those integers, those whole numbers. On something like this, which is a bit more experimental, you can get the in-between, which is great because sometimes we do want that nuance. It might be more meaningful. Activity tracking, for those who are able to do it. We sent 4 of these I don't have any on my desk, but 4 of these accelerometer devices to wear on people's wrists and their ankles. They're just like a little bit bigger than a smartwatch, maybe a little bit chunkier and a little bit more prisoner looking. But they wear them for 1 week out of each month on their 4 limbs. And they wear them as much as they can comfortably, not going in the shower, or maybe not sleeping with them. But that is what we call passive wear time or passive data collection. So they're not doing anything special. They're just living their lives. People will log maybe if they're doing like yoga or going on a walk or something like that. So we can assess sort of their natural living state as it relates to their like limb strength and limb movement. But because people do different things and we can't compare, if somebody does yoga all the time and somebody doesn't, then their data is going to be different. And there's ways to control for that. But we do have active wear time, which is where people on 3 days of that week, they'll sit down and do this little 8-minute video seated exercise session, which consists of arm lifts, leg lifts, and seated toe touches. And this provides us a nice standard to compare people's data, to each other, because most people are doing this part of the study. I'll mention again that some people don't choose not to do one of these 2. So maybe they'll only do the exercises, but not do the passive wear time, or vice versa. And that's okay. We would like if everybody could give it a try, at least, to see if it's feasible. But we want to make it as comfortable for people as possible. And this is a graph example of the exercise portion, the seated exercise data that is shown in the portal. The passive wear time is a little bit too large to show in the portal, though we do use it. So people will see their active wear time data after they send the devices back to us. And then the last aspect is the remote blood collections, which we started back in the fall of 2019. And we send mobile phlebotomists to people's homes roughly every 3 months. With COVID, it was much more difficult than that. And we've sort of finally recovered that. It's pretty standard to do 3 months now. But we partner with various mobile phlebotomy providers across the US and more recently in Ontario, where we get pretty good coverage across the US now. There's some areas where it's too rural, or there's just not enough phlebotomists around to send out, but we can get pretty good coverage across the US. And if not, participants have recommended local labs or groups they know in the area that they've gone to. So we really try to go and try to get everybody their blood sampling done, and in Canada as well. What we do with the remote blood collections, it's a lot. This is sort of the central dogma, I guess, of biology, but we do something at every stage of this progression here. So with DNA, we extract DNA to do whole genome sequencing and assess for the presence of ALS-associated mutations. It is, for us, we're a research lab, so we're not a clinical lab. We can't give anything that can be used diagnostically, but we will share with participants whatever we find. Our panel is somewhat large because we can do whole genome sequencing, but it may include things that are a little bit less linked to ALS than others. So for example, SOD1 is of course like a very strong link to ALS. C9orf72 is a strong link to ALS. Maybe something like Senataxin is not as well known, but we'll assay for it. And the good thing about doing whole genome sequencing is that if somebody discovers a mutation that is strongly linked to ALS in the future, we don't have to resample and re-sequence the genome. We can just go back. We have the genome. We can look for that new mutation later down the line. So it's great for one time. Like if you can give blood samples and do this one-time thing, it'll last. It'll hold value for a very long time. And then RNA and protein I kind of group together because it's super exploratory. It's going into the unknown of the biology of ALS. Like I said, there's so many gaps left in the field, but to try to figure out new pathways, new proteins, RNA missplicing events, things like that, we'll capture blood samples to look in like serum and plasma for changes in the levels, usually, of the protein or RNA involved. So hopefully this can either find something new for us to develop in the lab or validate maybe another researcher who has found something in their lab. We can look into our samples and see in our larger cohort how those changes or those protein levels or RNA levels are represented in our group. And I always try to sum up that part by saying it's very important to collect any of this data. Studies that are collecting any individual part or 2 or 3 of these is great. Of course, the power in helping us figure out ALS comes from all of it. And the other point I started emphasizing more recently is because we're trying to do so much now in understanding ALS, we want to be able to capture everybody's story accurately as we can and in as many ways as we can. If it comes that somebody, they discover a new protein, a new enzyme, that it's upregulated in ALS patients compared to healthy controls, which is great. That's a huge finding, and we want stuff like that. To go a step further would be to figure out, okay, what presentations did those people have? Because if we're going to develop a therapeutic, it might make more sense if it's only targeting this subgroup of people with ALS. And if we're not capturing the data on that subgroup of ALS well enough, we won't know who they are. And then we might muddy the clinical trial data by enrolling people who the drug wasn't effective for and missing the people who it was for. And if we're not having sensitive measures, we might be missing small changes so if there is something that even showed a sign in somebody's fingers that there was improvement, but maybe the ALS-FRS doesn't capture it, then we lose information. And it's, to my opinion, that we don't know enough where we can just lose that information yet. So we need to be able to capture enough data to describe somebody's ALS story. There's a point to be made about if it's only improving finger movement a little bit, is it worth, investing in that drug? I would argue we don't know enough about ALS that it's not worth losing that data. So that is the ARC overview. The other part of ARC, which John mentioned earlier, is the ARC Data Commons. So this is more for researchers. I won't go too much into this, but we developed a platform on Google's Looker platform where researchers can have sort of like buffet access to our ARC data. It is completely de-identified data. Nobody's going to have access to any of the participants' identifying information. And to date, we've just crossed over, I think, 100 different groups who have requested access to the data. And it's great because ALS TDI doesn't have the expertise in everything. So if there are speech pathologists or people who are specifically good with accelerometry, it provides an easy way for them to access our data. It also provides an easy way for like clinicians to access this, because we tried to make it easy enough that it doesn't require, knowing too much like code, or really any code, to download large data sets. So this has enabled us to collaborate more with more different groups of researchers. And it's, spoiler alert. It's been really exciting since we've launched this to see the types of groups that have come in and the collaborations we form, because it's more than we've ever done in the past. So this has been a really great platform. And again, I'd thank the participants for all the data they contributed to make the interest in this grow from other researchers. Okay, so I wanted to touch on highlight some of the collaborations that we're doing and might be coming a little bit. So on the sample collection side, we joined the Global Neurodegeneration Proteomics Consortium, which is a lot of words, but it's a Gates Venture backed program where different research groups from different disease indications, diseases, have contributed their proteomics data to a database where it's going to be harmonized. So all the data is in like the same format, to which the people in the consortium will be able to share and look at each other's data, compare across diseases, but then eventually to the larger research community as well. And I wanted to highlight a few other projects we're doing with Dr. Richard Bedlack at Duke. I think he released a publication recently, if anybody saw that, on IGF-BP7, which is a marker he found, it's both a protein and a gene, that he found in his ALS reversals cohort that might be linked to why they are reversals cases. So he asked us to assess the levels and presence, the levels of the protein form of this, and then the gene itself, the presence of the gene in our cohort. So we're doing that this summer. LifeArc is a group in the UK who has research capacity, but also is funding ALS research. We're looking at MMP9 and TIMP1, which are enzymes involved in a lot of things for ALS purposes in inflammatory response. So they funded our project to look at those levels in our participant cohort. And then we're doing a large-scale neurofilament light study assessing different assays that measure neurofilament light, but of course, seeing how those levels are in our group and how they are longitudinally, which is something that isn't super fleshed out in the research yet. It's coming around, but we'll be able to add our data to that growing field, which is really exciting for us. The surveys, so the personal background, if you remember, we were awarded a grant by the CDC last year to essentially assess all that information, all that survey data. We're working really closely with Danielle Boyce and Pat Dolan, who I'm sure a lot of you know, to both look at that survey data and the clinical data, as well as the geospatial data and the mapping of like where people have lived, and if they've lived near any sort of like environmental event that happened, or near farms, or places that might have high pathogen risk or anything like that. So that's another huge project we started this year. That's super exciting. This is from last year. This was the accelerometry aspect of our study. Dr. Gupta at MGH looked at that passive wear time aspect of our accelerometry data and published on it, basically showing that it is more sensitive than the ALS FRS, and that it captures progression a few months earlier in some cases in people who wore the devices. There is active, I didn't list it here, but there's active work going on for the exercise portion as well. I'm hoping maybe by the summit we'll have something to show on that. So we're hitting both the exercise types or the wear types for the accelerometry. And then last month, actually, I think, end of last month, or June, I guess it's August, right? Yeah. A team at MIT and Takeda had our voice recording data. And they set out to develop a model that was a bit better than the one we have, which they did, but also use that model to predict progression. So this is in preprint right now. I've put the link here, but you can go search it. So this is a nice step forward, next step for the model that we developed a few years ago, and hopefully will make that aspect of the ARC study, even better once it's, fully published and validated. I guess I can give you like a come to the summit, watch the summit. Hopefully you'll have more details on like any of these. I will talk about in more details at the summit. So I guess that's like an advertisement. I don't know.
We'll put a link in the chat for the TDI summit coming in October if you'd like to learn more.
Yeah. Or reach out to me if you're curious about any of these. I know I can't go into them in detail right now. Some other final like coming soon points I wanted to touch on. We are obviously continuing to collaborate with more groups than I mentioned, and get ready for more data harmonization efforts. I highlighted the Answer ALS collaboration we're doing this summer. Answer ALS has another large data set of patients who contributed a lot of different data. Their study ended, but we found an opportunity where we can try and join those. Requires some data harmonization because we collected a little bit differently the data, but it gives us the experience to also do that in the future if there are other studies similar to ours, and maybe we couldn't join up at the beginning, but with some planning we can try to stick as close to each other as possible, conduct our studies in parallel, join the data sets at the end, make the most out of everybody's data. Further biomarker testing and analysis efforts and reporting. I already highlighted the biomarker stuff and the protein stuff, so I will skip that. But if you're in ARC, you know that there isn't a lot of blood stuff that shows in the portal. It's just like basic blood work, but hopefully once we finish a lot of these larger studies, we can start reporting them. We have to be able to describe their clinical meaningfulness and things like that.
Alan, I'm going to interrupt you there just because it's pertinent. We did have a question asking about, are you following NFL? Yes, IL-2 and 14 and TDP-43 in the blood work.
Yep. Yeah, so not maybe all at the same degree. So for example, we've used SomaLogic SomaScan, which is they have SomaLogic is a company that does large scale protein testing, thousands of proteins. Those are included in their panels. Not every sample in ARC has gone to that yet, because that requires more funding, but we have done, I think, almost 400 samples of the 800 blood draws have gone to that. So we are, and we're going to keep trying as much as we can if we can get the funding. Yep. Developing new digital health measures and further testing of our existing ones. The existing ones I sort of already touched upon, but again, to the point of, can we capture as much accurate information about somebody's ALS story as possible? Do we need more measures? Can we validate them? Can somebody else maybe validate it and we test it in our protocol? I think that is a point of focus for ARC as well. Increase outreach efforts. So this is going to be a big one for us. Because we're a remote study, we have a unique opportunity to try to go to places that are hard to get, more I can't say rural places, or maybe other countries, to get populations that are, more diverse than ours. We mainly have people who are Caucasian, live in major cities, have access to good healthcare. Of course, there's more people with ALS who are not in that description group. So we want to make a strong effort to get ARC to more people in more areas. And then improve functionality and accessibility of existing tests. I'll be the first to criticize ARC stuff. There's like UI stuff, portal stuff that we can update that I think is not great. Maybe participants think is fine. That's just like make things better. I just throw that on there as sort of like a check for myself. Like we need to do more on that front. So that was a lot. That's like all the ARC overview, status of ARC. I'm gonna hard switch gears into introducing our two little guest speakers here, who are friends of mine, and thank you for being willing to do this. Gwen Peterson and Rick Zweep. I highlighted your names in pink for some reason. I hope you feel embarrassed or flattered. I don't know. the glow added
Flattered.
Flattered. Okay, great. So Gwen's been in the study for quite a while, so she'll be able to provide, a perspective of being a longtime participant. And Rick joined more recently. Gwen and Rick, I want to give you a quick chance if you want to say anything else about yourself to the people here before we jump in.
Yeah, I'm off mute. I can go really quick and more time for Q&A, so I'll be quick. Over the last 4 years, I have participated in ALS TDI's in-home blood collection program, which falls under ARC. I've got to say, this has been the easiest and most seamless research study I've participated in. I'm sure it's not cheap to send out in-home phlebotomists, but I've got to say it's really easy for me. I've heard from others just the ease of this study. So ALS TDI, thanks for making observational research, which is so important, so accessible and easy to people like me. So I'll shut up and Rick.
Yeah, I came to the party late. I went to the summit last year for the first time ever after like about a year after being diagnosed. And I'd heard about it, and I'd seen pictures of people with motion tracking bracelets on and stuff like that. And so when I finally sat down and sat in the portal and registered, I was just amazed by how simple it was to register and how transparent, particularly the transparency, because at that point I was still a bit paranoid about my data, especially because I live in Canada and Americans scare me sometimes. So, no, I'm just kidding. The fact that we could even participate from Canada was like a huge plus. And then since then, you've seen it in the presentation today, the transparency, right? Like to be able to participate in a study that's so easy, like Gwen said, and then to see where the stuff goes and that it's actually doing something almost in real time and benefiting maybe us and certainly future generations of people with ALS. It's like the easiest and most promising thing I can do as a person with ALS. So my kind of conclusion was, well, why wouldn't everybody want to do this? It's so easy, So that's my new little side mission, is to silently and quietly in the background recruit people.
And thank you both for all the compliments. I wish I could force you to say not nice things. Maybe I will. Let's see. But I did want to ask Rick, because you are newer, did you have any sort of uncertainty before you joined? And after joining, if you had to say just like one short thing about somebody who is unsure, like on the fence about joining, sort of what would you tell them?
I did have some uncertainty. Like I said, I was concerned about where does the data go and all that kind of stuff. I learned all about anonymization and security and everything else in biomedical research, so that was a plus. I was had done some other studies through my clinic before where, 28 pages of paper and you give blood and that's the last you heard of it. So it was kind of like, well, if that's all it is, maybe there's other people who will do it. I don't need to. But then when I found out about the entire scope of the program, and like I said, it was just so easy, and the portal makes it fantastic, even as a side benefit, to see your own progress or lack of it on certain fronts. It's just a cool side benefit. So I think I fall back to my previous statement, which is it's so easy. Why wouldn't you?
Mhm. Yeah, it is a point we try to make. There are there's definitely other studies out there, and it's hard, I think, for people, at least as I've been told, when there are multiple studies that are asking you to do multiple things, that's a lot of time to commit, and not everybody has that bandwidth to do it. But because we try to make this study easy and make the tasks optional, it's sort of easy to pop in, try it, and pop out if it doesn't work for you. So I'm glad that's something that you felt came across. And
you took away the
Alan-
fright factor, right? Because I didn't
Yeah.
have to do everything all the time,
Yeah.
but I still do. So go ahead.
Thank you.
And Alan, I had a question about that in chat. I asked, so I'm one of those people who is kind of inconsistent with how much I record, and when I do my FRS surveys, will my inconsistent nature negatively impact or have any impact on the data?
Mhm. Yeah, so I guess sort of being honest, it depends on how much data in total we collect. In some cases, that would be hard because maybe if we were trying to get like a certain range or a certain frequency of collections, we might not be able to use your data in that analysis. But thankfully, because we have so many people who are giving their data in so many different ways, we can sort of say like, all right, we need to do this analysis. Do we have people who submitted FRSs this frequently in this range and gave samples, which is something that you'd probably fit in, right? Like, so you would be using that. So it's not like maybe not in everything, absolutely everything, but there's definitely places for everybody's data. At least like 3 months, hopefully, or 6 months would be great of like anything, even if it's just 2 things. But there is a place for everybody's data. But I guess before we switch it over to the audience, I'm just gonna ask one more time. Do you guys want to say anything negative? Because I love constructive feedback. I will take negative feedback.
On your last point, Ellen, it's not negative, more constructive. It's it's good to know that consistencies in the data are favorable. And so with that, like, does TDI have any ask or any action of participants to, record more consistently, or like, what's the ask of participants?
Yeah,
If there is one.
So we try to word it sort of like, this is the ask is to do this monthly. And then like at the bottom, it's like, but if you can't, it's okay. So that's like the first way we do it. And then we have the email reminders that will stay on track, even if you decide that you don't want to do it that frequently. So it is a little bit of a poke for some people to get that if they don't want to. Of course, they can talk to us and we can try and shut those off. But we try to emphasize the fact that the more data, the better, and the more frequent, the better. We will mention that it's a little bit easier with the blood draws and the accelerometry because we're actually emailing each month, right? And maybe it's a little bit more pressure when they're talking to us. But for the FRS and the voice recordings, it's mostly just reminders.
Reminders are magnificently gentle. They're effective. Like they're not overloading, right? Like some things you sign up for, you just get overwhelmed with stuff afterwards. This is just the right kind of cadence and and the right frequency, and it works.
Well, thank you both for taking the time to do this and also for contributing data. I really appreciate it. You guys don't know, the rest of the people on the call, I love these two. They're awesome. Super cool people. I still haven't met Gwen in person for some reason. I think we keep avoiding each other.
Oh, you're kidding.
Yeah, I don't know how it happens.
I
Yeah.
know.
Yeah.
We will.
Yeah,
We
it'll
will.
happen. I do want to shout out Christine Moretti as well. She gave me some feedback that Gwen and Rick covered all of those points, but she's another super ARC participant user who wanted to contribute some feedback to this, but she shared a lot of her similar thoughts to Gwen and Rick. So with that, I guess I can turn it over to the chat questions. Hopefully there's enough time to do some of them.
Yeah, a bunch of them we've covered, Alan, so that's great. But
Love it.
one, Lynn had to pop off, but one of the things that he was saying is that his phone interface on his eye gaze computer is iffy and a web interface would be better. How do you guys engage with eye gaze systems and all of that?
Yeah, so that's a good point. He brought that up because I do think it is a weakness. We do adapt our pages to be like mobile fitting and tablet fitting. And some people, I don't know the exact numbers, but there's some people whose eye gaze technology doesn't impede them at all from using the site. But I've had a couple of people reach out saying that they can't hit certain spots because of the software. It just doesn't line up with our website. So we're trying to do what we can without this technology in-house to adapt to those situations. We love hearing that feedback because we don't know, because we don't have the eye gaze technology in-house. Ideally, we'd like to maybe get some users who are testing specific devices to help us troubleshoot. That has happened in one case in the past. But we will take feedback like that to try to work with people to make sure we can adjust things for everybody.
Yeah, that's kind of a question I have for Rick and Gwen and anybody else who's on here who's willing to come out of the art closet and identify an answer. Do you use your computer or your phone to participate in the program?
Voice recordings, phone, ALS, FRS, typically computer.
Okay.
Same for me.
And do you guys go in and look in your portal at your data? You do.
Not
Yes.
obsessively.
Yeah, not obsessively, but yes. And it's nice that I have the option of looking anytime.
Well, I think that's a big deal. I meet a lot of people who are like, I don't want to look at it. I totally get that. And sometimes their sister, people will come in for a tour and their sister's sitting next to them going, I'm a data person. I do want to look at it. Okay. That's up to you guys, but you don't have to look. But if you get into this stuff, it's pretty interesting to be able to look. So it doesn't draw you into rabbit holes, you guys. Good.
It's a really interesting question, Carol, because the FRS score is the one that really piqued my interest, because all of a sudden I realized, hey, mercifully, my score isn't declining as much as I feel like I'm declining. And that brings up thoughts and research about the validity of ALS FRSR and all that jazz, which we all know, about that. But it also gave me a moment of kind of satisfaction that, hey, I'm on a bit of a slow roll, and that makes me happy, even though I feel like I'm not. You know what I mean? Because there's that variability between the different levels of the questions where it's not captured.
Well, and what you
And,
feel matters too, right? You guys, I know that Alan and the clinical team added in, kind of mood and feeling emojis into ARC this year because that matters. You may have been depressed, right, Rick? Well, or upset and concerned and worried to look at it. And then all of a sudden, that can change. And it can also, I imagine, affect how you answer the questions on the ALS-FRS questionnaire. It's you're a human before you're a person with ALS, right? What were you going to say, Gwen? Sorry.
Sorry, no. And this is the only place I know of that's tracking my ALS FRS on a graph. So I I wish I could log into my chart and see it there, but I can't. So, graphically, this is the only spot I know of.
Yeah, it's pretty cool. Alan, also, I thought this was interesting. I think it was Lynn too, talked about the fact that the FRS barely measures upper motor neuron involvement. Can you speak to that?
Yeah. Well, I don't know if I'm an expert to be able to speak to that. But how those symptoms present is not indicative of what region of the motor sort of upper lower motor neurons that or actually just upper motor that is involved. So I don't know if we'll be able to capture maybe ideally if somebody could develop a technique maybe or a device or something to be able to assess those symptoms, that's something we would want to include because it's a gap. I guess it's a gap, and it doesn't tell the whole story, right? You can get some assessment of lower motor neuron function from what we have currently. With the FRS, and it's still even with what we have currently, there's still a lot of upper motor neuron stuff that we can't reach out to. So I'm not like a big expert on that portion of ALS. So I don't know what the field's doing as far as in clinical research, but whatever we can do to adapt it into our study is sort of game for us.
Is there anyone who's incapable of typing who would like to use their voice to ask a question? Happy to allow you to unmute or unmute you. Okay, anybody who can type and still wants to ask a question, we have a couple more here, but we'll make sure to get back to those people because some of them have left. So I want to be sure if there's one of you out there with a pressing question, make it be a good one, you guys. Anybody?
I wanted to say the other big benefit of participating that I hadn't expected is what we see here on this call, the fabulous folks from ALS TDI who are real people who really care, and they're approachable and they answer questions, and you can email them and you can just, it makes it so real and it makes such a difference.
Oh, that's so nice. Thank you. I mean, these are real people. Beth, would you wave your arms around too? And Danielle? And I don't think Erin's on here, but some of your ALS TDI clinical team is on besides Alan. These are real people. They will be the ones answering your emails. They will be the ones looking at your anonymized data and helping to share it with researchers across the globe so that we can't do this by ourselves. We think we're pretty darn awesome, but there is no way we can do this by ourselves. And you guys are creating, what are we at now? 36 terabytes of data. I don't know how long it takes to do an uptick in a terabyte, but that's an awful lot. And we don't have the money and the infrastructure to be able to look at that data and leverage it into treatments. There might be answers in there that we don't find and somebody else will. So you're not just helping us. Alan, I didn't even know that number of 100 people or 100 entities accessing the data. So that's really cool. Any other questions other than the fact that Gwen would like to be paid, Alan?
Oh, okay. Yeah, sure. Yeah. I did bribe them to do this, so I should pay them.
You did?
Yeah.
No, it wasn't. Carol, things like that help with recruit. Yeah.
Yeah, I don't know what effect that would have on an IRB that we've already tested, but
Yeah.
it's interesting.
I have no idea what that would entail, but maybe it would help with recruitment. Who knows?
It's something we should think about. And Danielle, you just popped something in there. You want to say it out loud?
Yeah, Rick had asked if we asked about falls, and I commented, yes, we have a whole section on injuries, which encompasses falls. It's quite detailed, and I'm actually just started analyzing it. So that's how I know all about it.
Thank you.
And I know our whole team has gotten a lot of questions on that publication of Rick Bedlack's, Alan, that you referenced earlier. And, you know, some of the people that you've heard from today are working on, you know, kind of a larger cohort from ARC to look at the levels of IGF-BP7. So stay tuned in the next 2 months and we'll see if we can see what we can tell you. Anybody else? We are over time. But I'm happy to stay on. If I know the answer to your question, I probably won't. But if anybody has any others, please ask them, and remember that you're going to hear from us and offer us feedback. Anybody? Okay. Thank you, everyone who is participating in ARC, everyone who is helping us to cover the cost of ARC, everyone who is executing ARC, and everyone who is part of the team that is leveraging the data for their own research. Really, really grateful.
Thank
Thanks,
you.
everybody.
Thank you, everybody.